For identification of mycobacteria, there are some couple of techniques you can use. These techniques are kind of like, let me say, ways in the lab that we can be able to use, okay, and try to identify a mycobacteria species. So let's quickly look at the techniques for identification of these mycobacteria. We have a couple of them, and I actually arranged everything in this lecture for you guys. So we have automated culture system, also known as such as BATEC. We have the nucleic acid probes with PCR. We have gas liquid chromatography, high performance liquid chromatography. Then I think that's all. So starting from the automated culture system such as BATEC, we said that what the BATEC, fully known as the Benton-Dixon and Company, is an automated blood culture system that uses fluorescence technology just to dictate the organism growth in blood culture bottles. So for you to use this BATEC, maybe you collect the organism into a bottle, right? And this BATEC is a system that will help to like check how the organism is growing, okay? So the BATEC blood culture vials, vials contain ingredients such as what? Soybean, casein, digest broth. Yeast, it contains amino acid, contains sugar, contains vitamins, contains sodium, contains the polyanethyl sulfonate, which is an anticoagulant, right? So this system will offer a continuum of care across the entire healthcare. Offers what across the entire healthcare system, right? Just trying to look at it, all right. But the main role of this automated culture system, such as BATEC, is just simply for you to just access the growth of the organism, all right. Then moving to the next one, which is a nucleic acid probe with PCR. Nucleic acid probe with PCR. Okay, sorry. Okay. This is nucleic acid probe with PCR, right? So that a variety of nucleic acid probes are available commercially as kits for dictation of microorganisms in infection in what human beings. So basically they are available so that it can help to like dictate, okay, the microorganism that is actually infecting a human being, right? So since the mycobacteria are also microorganisms, you can also use to dictate them. So some of this can also be used to dictate the same pathogens in specimens from dogs or cats. The probes are available for use in the clinical microbiology laboratory to identify organisms that have grown in the culture. Then going next to the gas liquid chromatography method, we said that the gas chromatography is a common type of chromatography used in analytical chemistry for separating and analyzing compounds that can be vaporized without decomposition. All right. So the typical uses of this gas liquid chromatography include testing for the purity of a particular substance or separating the different components of a mixture. So in that way now you can be able to separate the microorganisms that are actually in a specimen that you collected. All right. Then high performance liquid chromatography. High performance liquid chromatography, formerly referred to as what? High pressure liquid chromatography, is a technique in analytical chemistry used to separate, identify, and quantify what? Specific compounds in a mixture, right? So the mixtures can originate from food, chemicals, pharmaceuticals, biological, environmental, and agricultural specimens, right? Which can be dissolved into liquid solutions. So I think this last one, all this chromatography is just trying to look at how we can be able to identify these microorganisms and also purify them. Okay, because sometimes they might be mixed up with some other things and all that, right? So these are the methods or techniques for identifying mycobacteria.